the Genome Writers Guild Conference in 2020, coming to you virtually this year. So thank you everyone for coming, and I wanted to, session 7's titled "Supporting the Public Trust." And so why should the public support scientific endeavors in general? It's pretty easy to come up with facts and figures and explain economic advantages, health advances, and so on. But ultimately, the role of a scientist is to uncover new knowledge and then go on to share it. So to achieve that goal, we must not only foster public trust, but prove that we deserve it. So with that, our next few speakers will be discussing a few key steps in promoting this trust, establishing regulations, setting objective measures, and considering potential side effects. So our first speaker is Dr. Lluis Montoliu, and he's coming from the Spanish National Center for Biotechnology, as well as coming as a representative from our friends at ARIGE, a group from centered in Paris. And coming from these two places, he'll be talking about ethics and how we are developing public trust amongst the ongoing COVID crisis. Without further ado, Dr. Montoliu.
Thank you, Adam, and thank you, everyone. So let me just right away share my screen. So thank you for this invitation to Genome Writers Guild. I think this is very adequate to run this online conference. I'm here to discuss some of the issues related to genome editing, particularly promoting the responsible genome editing, as we would like to present from ARIGE. ARIGE being the Association for Responsible Research and Innovation in Genome Editing, which we created a couple of years ago in Paris, in France, as Adam was saying. So we have a number of aims that I will not be reading for you, but basically the overall aim of ARIGE can be said that all emerging technologies share common aspects to look at multiple scales, as Adam was pointing out in the introduction. The diversity of these questions in all these emerging technologies, they need a place to discuss. And in regard to genome editing, we would like to think that ARIGE should be the right place for addressing all these different questions, being in an inclusive place where all the different stakeholders will gather to discuss these different issues. This is the current board of ARIGE that I have the pleasure and the honor to be the president of, and together with my colleagues, and we are very fun and very proud of a very diverse scientific committee, including Stephen Ecker, which is also playing a decisive role in Genome Writers Guild. As you can see, we have members of the scientific committee from all over the world. So let me just put you back in November 2018, and this is when we first learned about He Jiankui from Shenzhen and about his irresponsible experiment of doing genome editing and applying genome editing on embryos and then transferring these embryos and leading to, as we know now, 3 girls in China. That triggered a first response from many different institutions, and we, of course, in the beginning of December, we presented a report, we presented a statement in which we basically say we should not be using genome editing on human beings, and then these techniques are deemed safe and effective and with precise therapeutic applications justified after a broad and open debate. And I'm bringing this on the table because a couple of months later, in mid-2019, we learned about other people, such as this Russian scientist, that he was willing to repeat the experiment. And that triggered that not only ARIGE, but this time in combination with Genome Writers Guild and the Japanese Society for Genome Editing, we promoted another common statement in 14 June last year, in which the summary of the statement was that this CRISPR genome editing revolution has a tremendous potential to bring benefits and cures to the human mankind. But its deliberate misuse can also bring uneasiness, anxiety, and mistrust. And as a society, we should stand against any attempt to use these techniques prematurely, dangerously, and irreversibly on human beings. And this is something that, as we know, ended up in a good way, I would say. China eventually condemned this person, this researcher, and his collaborators to prison, to fine, and most importantly, they were inhabilitated for life. And as you know, He Jiankui was not found guilty because of the genome editing experiment he did, but because he was pretended to be a physician without being such a physician. Nonetheless, he was condemned, and then we should be all happy about. Uh, Very recently, just about 1 month ago, we read in Nature about 3 different manuscripts that have been deposited in bioRxiv from Maria Yazin, Dieter Egli, Katinia Khan, and Shukran Mitalipov, again providing additional reasons why we shouldn't be doing this type of experiments, particularly for all the different types of deletions and reshuffling and reorganizations that were associated with this type of experiments. And this was a good reminder why it shouldn't been possible, or this experiment that was done by He Jiankui should have been done, and why it's still 1.5 years later, it was a bad idea to conduct this experiment. And it is because of these current limitations that we should be very clear when we're talking about the society. We should be very honest, and we should be providing the society with the current benefits and the potential applications of genome editing. But we should also, it is our responsibility, and in our reach, we do this very seriously, because this is about promoting responsible genome editing, these current limitations, which can be summarized in these so-called off-target effects, in which similar sequences can be targeted, and this is not in the plan. So we plan to promote an edition in a given gene, but there is a similar gene that might be edited as well. This is something that we can work out and we can improve. There are different ways, bioinformatic means, using different nucleases in which we can reduce this risk. But most importantly, what we have is the mosaicism. The mosaicism that happens after this double-strand break is being promoted by these CRISPR genome editing tools. Then upon promoting this repairment, either if it goes non-homologous enjoining, particularly if it goes non-homologous enjoining, or as we wish, if it would go the homology-driven repair pathway, then we would end up with a variety of different solutions. This is the outcome of the standard experiment. As you can see, every single line, in this case, these are mice, every single line, they have different sequences. And this is the real problem we need to be clear about, and we need to be presenting in the society. This is something that we can deal with very easily in the academia. And this is also we should be very clear about in front of the society. We have here 4 different founder mice. They are all mosaics. The second of them, they seem to be carrying the mutated allele we plan. This is the one we choose. The other ones we discard. We choose, we set up the corresponding breeding schemes, and we hope that these different mutations will be segregated in the next generation. And eventually we will be able to establish the colony and do the experiments. Of course, this is something we can do with mice and we can do with plants, but this is not something we can do with patients. We cannot pretend treating a patient and saying, we don't know about you, but we're sure that your kids will be fixed regarding the mutation that you're carrying. This is actually out of context and out of discussion. This is not prudent, not ethically acceptable. But this is at the fundamental, and that explains how different is the genome editing applications in the lab or in the real life. Real life meaning the hospital, when we focus in the biomedical applications. For the biotechnology or for the biology, we can segregate these out, and we can always, particularly with plants, we can always get rid of all these unwanted alleles and eventually decide to stay with the correct one. Well, this is happening in the biomedical world, in the biomedical landscape. both for the in vivo and particularly ex vivo. We don't have many examples, but we do, and I think we will be hearing about lately in this session, in other sessions. At the beginning of this year, we hear about this ex vivo treatment of very complicated and very difficult to treat cancers like myelomas, sarcomas, and melanomas, in which these T cells from these patients were removed. They could be genome edited. There's some genes that can be knocked down, and then new genes can be reintroduced. Or a few months earlier, we heard about the successes on sickle cell anemia and beta thalassemia, in which the application of genome editing, again ex vivo, on hematopoietic stem blood cells, we can reduce this repressor, and by reducing this repressor BCL11A, we could rescue this beta globin for this very severe disease, so these people can live without transfusion. So you wanted me to talk about what is the regulatory landscape in Europe. Well, unfortunately, in Europe, not much has happened since 25th of July 2018. And that was the sentence we knew from the Court of Justice of the European Union, in which they decided that genome-edited organisms should be considered GMOs. And not only this, but they should be associated with similar risk for the people and for the environment as any other transgenic organism, which means that they are not electable for exception. And then the whole European Directive 2001 and 18 should apply, which means years of reports and millions of euros to be invested, which basically means that most of the biotechnological companies, they will have a hard time to promote any of these genome editing products. Here what we have is whether this CRISPR should be considered transgenic. Well, everybody knows that if we add a transgene to a DNA, we will get a GMO, we will get a transgenic, so there's no discussion about this. Also, everyone will agree that when we're comparing different genes from different variants, different breeds, they might be different, and they are not considered GMO. So this is the variety that we're all using in plants and in animals. But what happens when we use the CRISPR tools to transform a given variant into another? So we can choose the very long route of following the breeding schemes, and after dozens of generations, we might be close to something like this, or we can do this in just one generation using CRISPR. The question is whether this should be considered GMO. I know in the US you're not, but in Europe, unfortunately, we should say that they are. So these are GMO, which means that we should be presenting all these different permissions and etc. And this is not only applies for the companies in the biotechnology world, this also applies for any research, or any research involving CRISPR is considered a GMO, so we need to provide the corresponding permissions. Of course, there has been a lot of contest from the researchers, so there is here a large number of institutions and centers that they gather together, they join force for enabling the use of genome editing for biotechnology. So far, there is no modification of the law, which is what we're trying to get. We're living in a global world, and of course, when we are blocking the progress in Europe, we're promoting our researchers to go elsewhere. And not only the researchers, but the companies. And you will see that some of the ideas or some of the experiments and companies that could be been developed in Europe, they will eventually be developed in South America or in the US or in Asia. So that's the way it is, unfortunately, at the moment. Let me finish up in the last couple of minutes by saying a couple of words with CRISPR and COVID. I think COVID has put us under threat, the whole world, but is also opportunity at least for three different things. First, as a reminder about ethics. I mean, we all want vaccines, we all want treatments, but we should remind the population, and I believe this is also the role of associations like REACH, remind the society that science has procedures, that we have timelines, that we have permissions, that we have protocols, that we need to do one step after the other, that we cannot proceed into the clinical essays without doing the preclinical essays, that we should be promoting efficient drugs and efficient vaccines, but overall, we should be always promoting overall safe research, safe vaccines, and safe treatments. Regarding CRISPR, as you know, this has been also used as a diagnostic tool, and we learned it from Feng Zhang with Sherlock and Cas13 in 2017. And as early as in March, Feng Zhang was promoting the first experimental use of CRISPR for diagnosing the presence of at least two genes from the coronavirus, SARS-CoV-2, with a sensitivity around 10 copies of the virus. Then we get itemerization of this Sherlock with Carmen, not the opera, but this new method in which Sherlock and microfluidics were combined that comes from the broad, in which over 1,000 different people can be diagnosed at once for coronavirus, or maybe 10, 15 people can be diagnosed for the presence of more than 150 different RNA viruses, which I think it's a very awesome achievement. We have also not only the Sherlock technology from the environment of Jennifer Doudna, we have the COVID-19, the detector, and the associated companies that have been used to detect, which means that this CRISPR, which can be faster, simpler, they are somehow less sensitive, maybe one log less sensitive, but they are qualitative and they are not quantitative. There is a new version of Sherlock, which also has been put forward from Feng Zhang, in which they aim to do 100 copies of the coronavirus. This is very simple. It's considered to be at the point of care. So this is something that was eventually approved in the 8th of May as an emergency use for the FDA. No, we don't know anything about this in Europe. So the EMA hasn't said anything yet. And the last but not least, what we have is this CONAN, which is another of these ingenious acronyms, in this case coming from the Cas3, for applying the same principles for detecting the coronavirus with CRISPR. So this shows how versatile are these genome editing tools, and this is also our role to transmit to the society, particularly when we are in deficit of having diagnostic tools. I don't know about the US, but in Europe, here we are now dealing with outbreaks all over, and controlling the outbreaks means applying diagnostic at the right time and very fast. And perhaps CRISPR can be of help in this area. Last couple of minutes and seconds, maybe, CRISPR as antiviral. So as you know, there is a new variant since 2018, which is called Cas13D. And similar to Cas13A or Cas12A or Cas13B, Cas13D, which is also known as Cas-Rx, cuts RNA, but cuts in a very highly specific manner. So it doesn't have these unspecific RNAs as the Cas13A. This was known since 2018, so this is very recent. And beginning of this year, Pandolfi was presenting this theoretical experiment. This is not a proper experiment. This was a proposal in which a Cas13D would be packed into an adeno-associated virus along with guide RNAs that are devised against some of the genes of this coronavirus. And the idea is to cut this coronavirus genome and thereby promoting their degradation. There has been from Stanford, as you know, in May, this was published in Cell. There is this new development, which they call Pacman, for prophylactic antiviral CRISPR in human cells. And they show for the first time that Cas13D can be indeed used for degrading this coronavirus, not only the coronavirus, but also the influenza virus, which is another RNA virus. Of course, the problem with this approach is that they started up from transgenic human cells because they had the human cells overexpressing this Cas13D, which is probably out of context and out of discussion in the real world in the clinics. But it really shows the potential of this technique. And I believe this is all what I wanted to say. And just to remind you who we are at the ARISH, and these are most of us at the ARISH board. And let me finish by just inviting you that on the 14th of November, we will be having this next annual ARISH meeting, which also will be online. Thank you for your attention.
All right. Thank you very much. And we have time for one question. We have one question from Dr. MacGyver. After editing a single cell human embryo, assumably there will be a sampling at the 4 or 8 stage to verify the desired edit. For inherited diseases, unaffected embryos will also be identified that can be implanted, alleviating the need for editing to begin with. So with this in mind, is there any rationale for germline editing of inherited disease alleles?
Well, at the moment, I think the risks are outweighing the benefits. And I believe these 3 new manuscripts that I referred, that we knew about from last month, they make it very clear. We're still not in control of what's going on. So in my view, and I believe that's the generalized view of my colleagues in ARISH, at the moment, we should not be promoting genome editing for inherited diseases at the human embryo stages, because simply the number of things that can go wrong, they're just too high.
And it looks like we have time for the other question, which is from an anonymous attendee. And it says, I'm in agreement that He-Jin Koo was highly responsible and unethical. So I wonder what if the experiment was performed such as to reverse a lethal gene mutation or suppress its effect?
Well, I mean, it is not the objective what we judge here. It is the device of the experiment, and that the experiment was done without taking into consideration all the different risks that we accept and we know how to deal with when we're working with mice. I'm using mice in my lab, and I know that 1 in 20 are the ones that get right, and I can discard 19 mice and I can follow my experiments with 1. I cannot transmit. It's not ethical to transmit this uncertainty to human beings. So definitely, I will not even discuss if the aim was instead of obtaining human beings that would be resistant to AIDS, resistant to the HIV, instead of this maybe curing a severe disease. I think this is not the point. The point is that at the moment we are not in control of what is going on when we are applying genome editing on these human embryos. And while this is happening, we should not be applying this.
All right, well, thank you. And so with that, we'll move on to our next speaker.
I'd
like to welcome Dr. Samantha Maragh from the National Institute of Standards and Technology. And she'll be telling us on how they're approaching taking developing new measures for genome engineering safety. Thank you.
Thank you. I'll share my screen. Okay, hopefully you can see that in full screen mode.
You are good.
So I'll go ahead and give you a bit about what we're doing at NIST. You may not know who NIST is, but we're supporting confidence in being able to utilize genome editing technologies. That was a great talk that we just heard that leads right into this topic. So for the purposes of this discussion, I will go ahead and give you a sense of what a genome editing process overview. So you have editing molecules. We've heard a lot about. There are different kinds of editing molecules. People should be able to use the systems that are going to do what they want them to do and have that flexibility. You've got to deliver the molecules in the cells. The cells might be in vivo. So they might be, if it's a multicellular organism, they may be there in the organism, or maybe you take them out of the organism and you edit them, or maybe they are the organism. Maybe you're a yeast or you're a bacteria. So your goal is to get a DNA sequence change. We just heard, and I'll reiterate that that's not always the case, and it's not always the change that you want. You hopefully get edited cells, and then you have to assess whether they're fit for your purpose. Depending on what you're trying to do with the cells, you may have different stringencies for how confident you need to be about understanding what you've done to these cells. So I think about this in these three categories. I'm not going to talk about tools to learn and tools to make. Sometimes the cells that you're using are powerful for research. Maybe they're making you some secondary product, and the cell that you engineered isn't your product that you want. And sometimes they are your end product. So here's some examples of end products. We just heard about this as well, that you've got, that there are agricultural products, maybe microbial products as therapeutics, maybe it's engineering a mosquito so that it can't carry a virus or a disease as kinds of end products. What I'm going to focus on is the gene therapy application of end products. So as I just mentioned, you've got ex vivo, maybe you're taking cells out of a patient, editing them and putting them back into a patient as a therapy. It may be that you are trying to package the higher bar is maybe your goal is you can't take something out of a patient, and you might not be able to take the cells that you want out. If they're brain cells, if they're cardiac or heart, you might not have the option of ex vivo, and you may only have the option of in vivo. And that's a higher bar that you're trying to package these editing molecules, get them into the person. They have to go where they're supposed to go, get into the cells they're supposed to, do what they're supposed to do there. So that's a more challenging application. And so there are disease applications in development right now and moving forward. Just to give you a sense of organ systems right now that people are developing therapies towards. So the slant of the rest of the discussion will be primarily towards the gene therapy application, but a lot of what we're doing at NIST is trying to be generalizable to using genome editing technologies, but the use case is really to support that gene therapy use case as primary consideration. So this is information that I pulled from the internet, from the CRISPR Therapeutics site, and notifying that here's an example of a gene therapy that's moving forward. This tiny date up in the corner says May 12th, 2020, that this particular gene therapy product by CRISPR Therapeutics has gotten this RMAT designation from FDA. So that is a regenerative medicine advanced therapy. So this allows this product to move more quickly through a process because it's addressing a regenerative medicine application. And so the overview of this process, so this is a real process that's going on, is they have a patient, they take cells out of the patient, they screen to make sure they've got stem cells, they use genome editing, CRISPR-Cas9 delivery via electroporation, and they then screen those cells, and those pool of cells are their product that then goes back into a patient. So this is a real world example of what this process looks like that's moving forward in clinical trials. Just for another example, we hear a lot about CRISPR. CRISPR is not the only kind of editing tool and system that's moving forward, that's in clinic, if we think about clinical trials and trying to be moved forward to clinical application and actually state of care practice. And so this is an example where CAR T therapy is also utilizing genome editing tools to make these more robust or more fit for purpose products, either for autologous cell therapies where cells are coming out of a patient and going back into the same patient, or even to make what would be like master donator cells. So allogeneic, where you're actually engineering what would be cells that could go to many patients. So these are applications that people are trying to move forward with that are advancing. And so now we come to, what are the challenges that this field is facing in making sure that you have confidence that you understand what you've done to these cells? So I just want to give you this sense that there are different outcomes. We just heard this, and I want to reiterate this. This is sort of one of these takeaways as to how we think about, should you move forward with something? How well do you understand it? So at your on-target site, the site that you intend to engineer, you might be trying to engineer a few sites. But let's just take this example. Maybe you have one site you're trying to engineer. At that site, you might have your intended edits. So those are the things that are going to be meaningful to you, the thing that you want to happen. You may also have unintended edits. These things may be they may be okay, maybe they're not, but you need to know that they're happening, and you might not have any edits happen at that site. In the same cells, you may also have, at other places in the genome, off-target editing. So this is something where we can think about this as changing the DNA in any way, anywhere in the genome except for your target. You don't mean for this to happen. So we call this unintended editing. And the key here is that you have to survey the entire genome to understand if this has happened somewhere. And once you look, you have to decide, look at the entire genome as your search space and identify what happened there, as well as what frequency it happened. Is it only that, 0.1% of all of the cells in your pool have this, or maybe 50% of your cells in your pool have this off target? So you need to understand what it is and its frequency. And the challenge is that having to survey the whole genome creates a challenge. These things do occur. And you might be able to mitigate them by your design. They don't have to, you can have messier designs that maybe have more potential for off targets, or you might have cleaner designs, but you may not have a lot of wiggle room depending on if you really need to make a therapeutic change at a specific location. You may be limited in the kinds of designs that you have available. So this is difficult to measure and quantify with confidence, but particularly if we think about the gene therapy use case, the FDA considers this as part of their safety assessment profile. So you have to do this. It's important to understand where in the genome something might have happened and what happened, to then assess how concerned you are, if at all, that this affects the utility of your cells for your purpose. So if we just take the example of one site, let's just pretend that we're just looking at your on-target site. You start off with some cells. One, you always have to understand what you're comparing your measurements to. You're not actually editing the reference genome that might be, on NCBI. Your cells have their own genomic characteristics. They're from a different individual, not a pool of individuals like the NCBI reference genome. So when you look at your off targets, when you look at your editing, you have to say compared to the cells I actually started with. So that's a really important point. So you do the genome editing, just looking at the on-target site, you may have this pool, edited, unedited, and you may, in the edited, desired and undesired edits. So when we look at this a little bit further, there's some common challenges that you have to be aware of when you're thinking about this, and how you have confidence as to whether you understand what you've done to these cells. So you have this pool. You subsample some of this. You can't use all of it, or else you would be using up all of your product. So you subsample some of it. And then in general, just because of state of the art, and you don't usually have the opportunity to do single cell measurements on all of these cells, you're usually blending up these cells, extracting DNA, and then looking for the profile. And this is just a very, very small example of the heterogeneity that you see at a single location in the genome. You could then see this similar heterogeneity at off-target sites in the genome, wherever those happen to be, right? So when you look at this, it's heterogeneous. You may have limits of detection depending on the technology you're using to do these measurements. So say Illumina short read sequencing has certain limits of detection. Other tools might have different limits of detection to say, could you see 1 in 100? Could you see 1 in 1,000? What is that? Intracellular zygosity. So what happens in one cell on the allele from mom and allele from dad are likely going to be different. So you can't necessarily pull out information like dose from something like this. You might not know if you see half, let's just say, 50% of all your sequencing says you've got a variant and 50% says wild type. You don't know if 100% of your cells are heterozygous, or if 50% of your cells are homozygous, or something in between. So that's just one thing to think about when you're looking at this. DNA variant detection bias. So it's not necessarily that whether it's a SNP, something small or something large, a tool may not have the same ability to resolve that kind of DNA change. And bioinformatics pipelines, so one thing is that the reason that I believe that this is what it is because whatever came off of the sequencer went into an algorithm, and the algorithm spit this back out. How do we know that algorithms are giving similar information? So these are just some of these complexities. And so this is the image of, you might have very large changes in the genome, which might be particularly concerning because, you could just think of the more things that you rearrange, the larger amount of DNA that you change, maybe that's particularly concerning. So you'd want to also utilize tools that might be able to give you information about that kind of change. Okay, so collaboration on standards can move people. Everybody's moving towards this, but if we can move together in the same direction, working together, we might be able to get there faster for community needs. Okay, so how confident are you that your answer is correct? What's the consequence if it's wrong? And then what can be done to understand if the answer is correct? So here's where we talk about standards. Standards aren't a dirty word. Standards are enabling. They don't tell you what you have to do and lock you down into something you don't want to do. They're there to help you understand if the data that you have, you believe it, if you should make important decisions based on the data that you have. So this is really meant to be enabling, not limiting. And so NIST, National Institute of Standards and Technology, we're the government agency that exists to help in this space. We sort of think of ourselves as industry's lab when technologies are transitioning to industry, or maybe they're already in industry and there are some challenges. This is really where NIST helps work with stakeholders there, also in academia, also in other government, to help with making measurements or standards to help with different areas of global economy. So here's just a sense of what we do. There's basic research happening. Mostly NIST interacts here once these things are being translated into industry. And in this case, when we're talking about things that might affect public health and safety, that might be the outcome for this particular use case. Here's sort of an alphabet soup of different organizations that we currently work with in a lot of our biological work. And so I want to just give you a little bit of sense here from FDA. NIST and FDA developed the slide together, particularly FDA CBER for Center for Biologics, to describe what NIST does, our role, and how we work with FDA to support their role as a regulatory agency in biological materials. And so we are not regulatory, FDA is, and what NIST can do is leverage and collaborate and foster, bring people together to understand needs, try to develop solutions, and taking into account what those needs are also for FDA, and work together so that we can come to solutions that meet FDA's needs, meet the stakeholder needs, and so that hopefully technologies can advance more rapidly and with more confidence that you understand the data and the reliability of the data that you're making important decisions on. Okay, so if we come back to this, what is NIST doing in genome editing? Well, I'm just going to put these blasts up here. I'm not going to read them. We've surveyed the community to say, what are the kinds of things that you need measurement help with, and see where that might align with what NIST can do. And so we've got all sorts of things, from algorithms to, how do we know whether the reagents that you're using to engineer the cells, that they're good quality, benchmarks for later down in the process. So there's a wide variety of things, and we're just starting to deal with a small subset of these, because you have to start somewhere. We want to be able to make traction and then continue to support the community. So how is NIST helping? I'll briefly tell you about this NIST Genome Editing Consortium, which is one activity working with stakeholders to collaborate on making solutions to pre-competitive needs that everybody has in the community. And we're also working on off-target assays. So if you think about what we do with the consortium, that's develop technologies and capabilities that when used for genome editing, there might be some challenges in understanding whether you're getting the right answer using those established technologies. When we think about developing measurement capabilities, so an example is off-target assays. These are things that are in process, they're coming out, they're new. How do we know that these new things are working well and that new technologies are reliable to deploy? How can we help with that? So there's one particular example I've highlighted here of work that we're doing with the NIH Somatic Cell Genome Editing Consortium, collaborating with St. Jude. And I have this other box that's empty of future work that we're thinking about, and where can we help? Maybe that's in what we just heard about, which is molecular diagnostics using genome editing that's expanding. How can we help foster confidence in tools so they can get to practice, maybe faster? So here's sort of how we think about what NIST does. We think about physical measurements, we think about data, and we think about documentary. How can we help with all of these things? So if we look at this process, there's a lot of innovation happening along the top here. What's underpinning all of this are measurements, the data that you generate from those measurements and the metadata, which I'm defining as the description of how you did the measurements or how you collected the data, how you analyze the data, and how you're housing it. So that's really what makes this transferable if somebody else is meant to get understanding, or even within your own organization, as people leave, people move, how do you maintain understanding and utility of the data and information? So I'll tell you about the consortium now. Take a few minutes to do that. So we have this NIST Genome Editing Consortium, and the goal is to work public-private, work with the stakeholder community, work with the genome editing experts to foster confidence in utilizing genome editing to make in research and primarily to develop commercial products. So it's divided into three working groups. One is really on the physical measurements. So primarily focused on DNA measurements, confidence in that. Data and metadata. So you have this data. In order for somebody else to get use out of it, or even you need to understand what exactly the study was that was done and be able to re-access that data to utilize it. And lexicon, so standard terms. There's no standard definition for genome editing or gene editing or off target. So could we help develop that so that there's better chance of more efficient communication and less possibility of confusion when sharing information or data? So our consortium members as of today, we have 37 member organizations that are all listed here, and there are a variety of different kinds of organizations. So for working group 1 in this consortium, so you design and execute, that's going to be different for your different purposes. You may do something like an off-target assay. These are really still developing, and there are lots of things coming on board. It's not really ripe for what the consortium does. And then we got the information that, that heterogeneousness that happens that doesn't actually happen in nature. It's because you're applying genome editing tools to a pool of cells that you get this profile that you would never see in, say, something like cancer diagnostics, where you think of, oh, there's a lot of mutations there. They don't all happen at exactly the same place in the genome. So the tools and the sequencing tools, the algorithms were never benchmarked to be able to find that. And so there's actually a challenge here in knowing for sure that the profile you get from that sequencing or that DNA analysis is actually a readout of what's happening at the biological level. So we're thinking about control materials, and they would plug into the process right here. They're not going to tell you about these upstream steps. You're using it to confirm your ability to appropriately read out the heterogeneity at the DNA sequence level. So I sort of already gave a sense of this, so I won't go over this. You can sort of read it as it's up on the slide, but if we have these control materials that could be shared, you could be able to better compare between different sites, know whether your instrument or your user is getting similar results over time, if you can combine data sets from another site. And this would really address the identity examples of different kinds of sequences, and that you can get different frequencies of sequence if you blend DNAs together. So here's the concept. We would have some cells that we would characterize the baseline in many ways. We would develop targeted assays, and that we would actually develop a series of engineered control cells and associated DNAs that these could be blended together to recapitulate not an exact example, but have benchmarks of something that say, I know that there's a 1 base pair deletion here and a 100 base pair insertion here, blend them together at frequencies and see, do you really pull back out that same information that you actually know you had those. So we have these 2 processes in progress. We're doing mixture studies and cell line engineering studies. So this is the concept of, if you blend different people together, you might identify sites across the genome where you have examples of everything from single base substitutions to mega base differences. And then you can ask, could your assay actually tell you that that was there? But that's not really the real case that people have in genome editing. It's really this case. You have a single site and a lot of variability at that site. And in order to do that, we're actually engineering cells, qualifying them for this single site, and then saying you could blend them together to generate a complexity profile, and then ask those questions. Do you actually get the right answer given a measurement, given an algorithm? So I'll briefly then tell you about this data and metadata working group. And the concept there is what are the data we're generating? How do you collect it? And how do you store it? So you want to be able to use the data that's growing.
Your time is about to be up.
Okay, great. So we have these we're looking at trying to say, how do you store this information in a way that could be transportable, usable, that people could actually populate? And then I'll just tell you that we have this metadata, this terminology standard that's going through the International Standards Organization right now. All of the terms on your screen right now are in development, have developed definitions for, and are in progress to become international standard definitions that have been vetted nationally, internationally, not just for human, but for other organisms as base general conceptual definitions. And there'll be much more to do as we think about trying to move towards therapy. So I'll just leave my thank you slide up.
Thank you. Thank you, Dr. Maragh.
Good morning. Thank you very much, Adam. I'm going to talk a little bit today about the accreditation process that is in place for cellular therapies, and that this will be able to apply to the complex processes that the clinicians face in trying to actually deliver these products to patients in a therapeutic manner. All right. So I am the president of the Foundation for Accreditation for Cellular Therapy. My term is just about to end. That organization, in fact, was organized in 1996 with the goal of raising the overall level of care in bone marrow transplantation, which is a model of highly complex care delivery in the clinic. These are some of the objectives, I hope, by the end of this presentation. And you've heard a little bit about why standards, and that standards is not a dirty word, and why accreditation, and that's also not a dirty word, because in both of them, they are facilitating the excellence of care to patients, and that is the ultimate goal of all of these processes. And one of the reasons for having standards and accreditation in the clinical area as well is it has now become an extremely rapid evolution of sometimes high-risk therapies, and we are always amazed to see how long it actually takes for skills to be introduced into the clinic. And 3 to 5 years is actually a relatively fast time, but now we must insist on these skills being implemented in a matter of weeks and months as new products come on board that are very different than anything that have been delivered in the clinic before. And that these also require large comprehensive teams that must be closely coordinated. And as I mentioned, it is really the bone marrow transplant world that is the model. Accreditation gives patients confidence. It actually also gives insurers confidence that the organization is qualified and able to deliver highly complex care. And then for groups that are trying to do multi-site clinical trials, the accreditation of the clinical team gives them some confidence that they will be able to handle the products that are being distributed by industry or by investigators at various institutions. So we're talking specifically about immune effector cells, and that this is one of the sets of standards that FACT has. And they are broadly defined as you indicated there, cells that have been differentiated into a form capable of modulating or effecting a specific immune response. And CAR T cells are certainly involved in these. Just for historical purposes, for people who are involved in bone marrow transplantation, donor lymphocyte infusions have not been included, as these are some long-standing immunologic therapies that have been kind of grandfathered into usual practice. So the core of standards is patient safety, improved outcomes, and access to these clinical evolutions. And the FACT-accredited transplant programs have been participating in immune effector cell trials, and there is a desire to apply FACT requirements to these new services. Drug manufacturers need some assurance of proper handling of their products, as many of them have high levels of risk at this point. Regulators are interested in the field's ability to handle toxicities, and payers are also quite interested in outcomes and expecting good outcomes for their covered services. So one question we often get is, my institution accredited for CAR T cells? And the answer to that is no. Accreditation from FACT is awarded to a program within the institution. And if another group decides to deliver similar therapy, it is not automatically included in that accreditation. And we're starting to see, as these therapies are proliferating, that there is great interest in other parts of institutions to deliver this care, and sometimes without the preparation. The bone marrow transplant programs have had, and individual immune effector cell programs. FACT has three main sets of standards. There is that apply in these cases. First, the common standards are a set of core standards that are common to any type of cellular therapy, and they are all included in the other standards. There has been immune effector cell standards were recently implemented, and they apply to programs that only perform immune effector cell therapy and not blood marrow transplant. And then there is an overarching set of standards that are by FACT and JC, our European colleagues, that they're the international standards for hematopoietic cellular therapy product collection, processing, and administration. And these actually include immune effector cell standards in those. There are different ways to organize care for immune effector cells, and I think we're going to see these evolve rather rapidly in the very near future. It had an opportunity programs that have conducted but as they proliferate, the patients being treated and the diversity of patients will probably demand different departments separate from the transplant service performing this therapy. But up till now, there are very few of these that have actually come into being. Applying for accreditation, there are transplant programs that administer immune effector cells that must comply with the requirements, and it does importantly include three major segments: the clinical care, the collection of the cells, laboratory processing of the cells. Specifically, FACT does not accredit commercial manufacturers, despite the requests that we've had from a number of new biotech firms. Three components, clinical, collection, and laboratory, can be accredited all together, or they can be accredited separately where there are some of the processes and procedures are contracted out. The several immune effector cell clinical models that exist, primarily blood and marrow transplant programs right now, But now non-transplant clinical sites, including leukemia or lymphoma services, are getting big enough that they may develop their own programs more completely. Sometimes it is delivered within primary immunotherapy divisions or institutes, and so these are several models of the kind of care. Very importantly, the relationship among the different clinical units is important. Some institutions have both a transplant program and a separate clinical unit that administer immune effector cells. They can have separate accreditation, or they can share accreditation if they share leadership, quality management programs, and staff training programs. And this just shows graphically some of the relationships among the units, that they may share personnel, bone marrow transplant nurses, or there may be separate, they may share physicians, but have separate nursing personnel teams that to deliver the care. There may be shared nursing resources where the bone marrow transplant nurses take care of both hematopoietic progenitor cell patients as well as immune effector cell patients, or there can be a mix of sharing of several different personnel groups across the services. This is a diagram showing an immunotherapy institute where immune effector cell patients may be across a broad range of units that are being cared for. The collection facilities are a specific part of the accreditation, and they need to meet the collection requirements of the standards. And it includes staff training, it includes knowledge of hematopoietic progenitor cells and immune effector cells. And that it's important because collection of these cells is sometimes carries significant toxicity. One of the biggest risks, in fact, of collecting mononuclear cells for immune effector cell manufacturing is actually the placement of the central catheter for collection if that's necessary. So it's just important that all of these components flow smoothly for the safety of the patient. The processing facility, the laboratories that manufacture immune effector cells, if these are on site, that at the institution and not a commercial company, then the accredited transplant program has to have a relationship and an agreement with that manufacturing facility. But the processing facility can apply also for independent accreditation. The IEC specific clinical requirements include requirements that are common to any cellular therapy, but have also specific knowledge, require specific knowledge of some of the newer syndromes, such as cytokine release syndrome, although now we're realizing that happens in bone marrow transplant too, as well as the coordination among all the different departments. And that as these standards evolve, they will evolve with the, as the toxicities and the management of patients related to these newer cells evolves. Working with third-party manufacturers, it's important for the program to verify that they have appropriate regulatory oversight, and they must qualify third-party manufacturers and define the responsibilities between the IEC program and the outside manufacturers. This does not apply if it is a commercial manufacturer, but if these are programs, manufacturers that hold INDs and biologic license applications, the clinical program is not responsible for verifying the GMP and GTP components, but this is different than the holder's ability to meet minimum requirements of the clinical program. The cell therapy is very much a team sport, and there must be confidence that they can work well together. So an example of FACT-accredited facilities using a licensed IEC product, the clinical program may perform vendor qualification as is done with almost any medical vendor, and that there can be a definition of responsibilities that must meet the minimum FACT standards. The apheresis and processing facility defines its relationship with the manufacturer, and then the manufacturer returns the products to the clinical program for administration. There must be infrastructure to manage different study requirements. It's in many ways quite similar to blood and marrow programs, but there are distinct differences where the products are shipped off-site, and it's important that there be methodology for maintaining the identity of the cells and knowing exactly how the cells have been handled and who's going to be handling them at each step of the way, and how that is coordinated. And the long-term follow-up is a very long-term now for these kinds of cells. So with each type of cell product, there has to be a system to clinically order it. There may be collection needs, and they are certain parameters that the collection has to qualify. The training for the toxicities and the potential side effects of each type of cell has to occur, and the programs need to do regular auditing and reporting to each other about all of the events that are occurring. I mentioned chain of custody, which is extremely important to make sure that the product is, the identity of the product is known all the way through the process, and that the conditions are met with shipping so that the product comes back to the patient. And this is all controlled through the appropriate quality management system. So there can be different documentation, but outcome data and reporting to each other is important as part of the quality management process. And the quality management program assures that the proper training is in place for all groups. So what is, with FACT accreditation, a group comes and of people active in the field. That means that they care for patients, they do the manufacturing, they do collections, and afterward, there is a committee that reviews the report of the inspectors, and there may be questions asked of the inspectors, and then it's this separate accreditation group that actually decides on whether citations should stand or not. And then a letter is sent to the program letting them know of what shortcomings may have been observed. And after a satisfactory response to those citations, accreditation may be awarded. Right now, everything has a COVID footnote, and that is right now that FACT has moved all accreditation back 6 months. That is, they've been extended for 6 months. We're studying virtual inspections, and upon restarting, whatever version of standards is in place at the time will be what the program is.
Your time is about to be up.
The 8th edition shortly. As of June, there have been 83 accredited programs for IECs. There's one standalone. There are accreditation in 4 different countries right now that have been for FACT. And we have 74 clinical inspectors. New standards will be out in March of 2021, concurrent with the FACT JC standard publication. And with that, thank you for your attention, and appreciate the opportunity to present today.
Thank you. Thank you very much. And with that, we are out of time for questions, so we'll move those on to the breakout sessions as well. In the meantime, I'd like to introduce Dr.
Great. As Adam said, I'll be talking about the ethical implications of incidental enhancements in human genome editing, and I'll be going through some cases from the literature. So during my talk today, I will provide you with several key definitions for understanding the cases I present. I will then discuss 3 specific gene cases from recent literature and describe how they fit into a framework that includes the potential for incidental enhancement. Finally, I will conclude with my assessment of potential challenges that policymakers face in the near future and address key principles that should be included in future governance. So for our working definitions, to start off with, human genome editing is the replacement, addition, or deletion of a single gene or set of genes in somatic cells or embryos with the intent to change and likely improve an organism's original genetic makeup. Beneficial human variants are variations of a gene that may randomly occur in nature and that may confer some socially desirable benefit to a person or organism that has the variant. For example, strength, inability to feel pain, or speed. And then finally, incidental enhancements are the potential for intentional genome editing to produce additional unintended beneficial changes that increase the recipient's social desirability. With those working definitions, I will now begin to discuss 3 gene cases from examples I've chosen for today. The first case is myostatin, or the MSTN gene. MSTN normally produces myostatin, a protein that is involved in regulating muscle growth. Myostatin inhibition can cause muscular hypertrophy and increased strength. A case study presented back in the early 2000s in the New England Journal of Medicine offered that the inactivation of this gene could be a potential therapeutic option for humans with muscle wasting diseases, such as Becker's and Duchenne's muscular dystrophy. More recently, scientists have used gene editing technology to introduce MSTN mutations into beagle embryos. Through CRISPR editing, the scientists were able to create site-specific genetically modified dogs. I have an image from this paper here on your slide. And the stark results of this gene editing are obvious. On the left-hand side, you can see the normal unedited beagles. So just for your reading purposes, these are hind legs and thighs. So we're looking here at the muscle density. So on the left are the unedited version, and then on the right, they did CRISPR into embryos of beagles, and you can see the increase in thigh muscle mass in both number 5 and number 11 here. The beagle research proposed that these results may not only greatly facilitate the generation of novel dog models for biomedical research, but also promote the creation of new strains of dogs with favorable traits for other purposes. This example highlights a potential for enhancement uses of MSTN. Moving beyond the enhancement of MSTN modification in dogs, human self-experimentation for enhancement purposes has also been attempted as recently as 2017. Additionally, the WHO Human Genome Editing Framework for Governance, which was released earlier this month, also refers to similar genes and their use in increasing muscle mass. This topic continues to have relevance on an international scale. For my second case, I'll be discussing the FA gene. FA is a gene that normally contributes to the regulation of pain, sensation, fear, and anxiety. A FA variant called FA out was recently discovered and discussed in a case study in 2019. And this variant results in an unusually complacent disposition and pain insensitivity. As I mentioned, the recent case study attributed this beneficial variant to one woman's remarkable painless recovery from an orthopedic surgery. In post-op pain scales, she rated her pain as 0 and 1 out of 10, when typically patients would rank it higher, much higher. This case report also comments on the patient's cheery disposition and self-reported little to no feelings of anxiety. Scientists have attempted to replicate the FA out gene variant in mice through the use of CRISPR technology. In doing so, they found that mouse modeling of FA loss of function mutations and pharmacological inhibition studies have shown a range of phenotypes, including hypoalgesia, accelerated skin wound healing, enhanced fear extinction memory, reduced anxiety, and short-term memory deficits. The research has proposed the potential for gene therapy that inhibits Fa as a means of treating post-operative pain, as well as chronic pain disorders, although this type of therapy has not yet been developed. In terms of enhancement, it's possible that the mellow low anxiety effect and pain and sensitivity that accompanies Fa out could potentially be targets that are socially and professionally desirable and beneficial. This would be especially true if targeting Fa could prevent a chronic pain disorder from developing or even elevate someone's mood in the long term. The first two cases I've discussed, MSTN and Fa, highlight the first policy challenge I would like to bring forward. Whereas both Fa and MSTN research suggested potential uses in medicine, specifically for MSTN, it had potential use in wasting diseases, muscular wasting diseases such as muscular dystrophy, and Fa had potential use in treating chronic pain syndromes. There's also research to suggest that these genes could be manipulated for enhancement purposes. These incidental enhancements could include increased strength in otherwise normal individuals who receive MSTN gene therapy, as this was demonstrated with the Beagle study, and increased complacent affect and pain tolerance, which has been demonstrated in mice with Fa knockout mutations. We'll need to decide as a society what boundaries we're willing to create in terms of how this genome editing can be applied, especially in a time where technology such as CRISPR has made it more efficient to introduce such edits. The final case I'll be presenting today is a cautionary tale. This is the case of actin, or the ACTN gene, which has several known beneficial variants. Specifically, the ACTN3 variants have long been associated with increased rapid muscle twitches and powerful muscle contractions. ACTN3 variants are one of over 250 single nucleotide polymorphisms that affect gene regulation and have been linked to athletic performance and increased speed. Beneficial variants of shortened actin-3, including the XX and the RX variant, have been found in a number of professional athletes, from sprinters to soccer players, tennis players, etc. This gene has even gained a lot of attention with at-home gene testing kits, such as 23andMe. Promising studies back in the early 2000s argued that the XX variant of this gene had an increased shift toward oxidative metabolism. There was also increased intrinsic endurance performance with some ACTN beneficial variants. This early research also suggested that these ACTN variants could be potentially exploited for gene therapies or prevention in muscular wasting diseases, again, such as muscular dystrophies, or prevention of sarcopenia in the elderly. Our knowledge of these beneficial variants has led to an ongoing discussion of the potential for gene doping in professional athletes and ethical implications of regulating this technology. But as I mentioned before, the ACTN gene example is the cautionary tale. It brings up the policy challenge of what do we do when our scientific knowledge advances and we learn more about the potential side effects of manipulating these genes. Specifically, while initial research in the early 2000s proposed ACTN variants as a potential target for gene therapies in human muscle wasting conditions, such as the muscular dystrophies and sarcopenia, more recent research has raised concerns about the unintended consequences of altering this gene, and continued research has called into question the validity of some of the earlier studies. It's possible that these so-called beneficial variants can come at an unanticipated cost. Specifically, a recent research study on female ultra-endurance runners showed that women with the XX variant had increased muscle damage, as indicated in the figure shown here. You can see that the pre-race versus post-race findings. In the post-race findings, there were increased levels of both myoglobin and creatinine kinase in the XX variant compared to the RX and RR normal variants. And this evidence shows that editing this specific gene may lead to, again, unintended severe consequences as a result of the high levels of oxidative metabolism. This new research raises additional policy challenges that must be considered when addressing human genome editing. Specifically, how do we propose regulation recommendations in the setting of our incomplete knowledge of these beneficial variants? And while there may be true medical applications of genes and genetic therapy for muscle wasting diseases and sarcopenia, this example highlights the potential risks of using gene editing for gene doping and enhancement in healthy individuals. As we fine-tune our understanding of the CRISPR-Cas9 tool and increase our fount of knowledge pertaining to specific genes and variants, the need for clear, responsible policies and guidelines grows. There are several challenges to incidental enhancement that have arisen with the increased prevalence of genome sequencing and new gene editing technologies such as CRISPR. In order to address these challenges, we'll need to anticipate a surge in our knowledge base of identified, defined beneficial variants. The 3 examples provided today offer potential medical applications for gene editing based off of previously identified, randomly occurring beneficial variants. Each example also hinted at some form of potential enhancement if the genome editing were to occur in healthy patients. For example, increased muscle mass, pain insensitivity, complacent affect, and increased speed and endurance. Rarely do researchers offer up the potential enhancements as a means of future directions for their research. One must look critically at the work they're doing and try to understand how this work could be applied to human traits in the future. As a society, we will need to decide where we are willing to draw the line when it comes to genome editing. So much of the current policy discussions have been focused on germline editing regulation. These cases have shown the potential uses for somatic genome editing as well. Do we agree on the boundaries of somatic genome editing? That's something time will tell. Finally, we must ask ourselves, who is best suited and qualified to govern these policies, and how can policies be enforced on a global scale? The case examples given today shed light on only a fraction of the genes currently under research investigation for the potential use of improvement of human health and quality of life. With the emergence of CRISPR technology and site-specific gene editing tools, we'll need to think critically about our societal values and strive to make a balance between scientific advancement and ethical innovation. I'd like to extend my gratitude to my colleagues at the UNC School of Medicine, the Department of Social Medicine, and the Center for Bioethics, as well as the NIH for providing funding for this ongoing research. And I'm happy to take any questions at this time or during the breakout session.
We do have time for a few questions. So I'll get started with one question. In terms of, you said that a lot of researchers don't report the potential enhancement traits that come with their research. Do you think that that would be a good thing to start providing for better consideration in the future?
I think it can be helpful in some aspects, but it can also maybe be a little bit dangerous in some aspects. For example, with some of the rogue scientists we've already discussed completing their own gene editing for enhancement purposes, it's possible that suggesting future enhancements might provide some ideas to people that may not be using it as responsibly as we would like. I mean, certainly in terms of identifying areas where policies can be made, it is helpful for scientists to kind of think creatively and outside of the box in terms of where enhancements could take place.
All right. If there's another, we have one other question from you. We have from Scott MacGyver, is enhancement necessarily bad?
Right, that's also a great question. I think that enhancement brings up a lot of course, ethical questions. Who gets to benefit from enhancement? What kind of enhancements are we okay with? Are we okay with the physical enhancements that I kind of alluded to in my talk with increased speed, increased muscle mass? Are we okay with cognitive enhancements, increased memory, increased learning capacity? I think there is a lot more to unpack with the term enhancement and the different ways that we can go down that path. But I'm not going to say that enhancement is all bad and none of it would be okay. I just think that there's a lot more to it.
Excellent. And with that, I believe we're at our time. So we'll be moving on to breakout sessions here. Thank you to all our speakers for this session. It was really fantastic to hear from all of you. And I think these are excellent ways to end up building public trust as we've